let's see, probably about 5 times as many people here tomorrow, or no, Wednesday at this time? Maybe, possibly, not tomorrow. If people are looking for extra questions, office hours, etc., just please let me know. Send me an email. Generally, tomorrow morning is relatively open. Other than that, it's going to be really quite challenging. My apologies for the lecture recording from Friday. Some of you may have noticed after about 15 minutes the sound died. I thought that I'd put new batteries in, but I guess I'd forgotten to check that it was turned off after last time. So now we're going to have also an interesting technological issue because I'm recording it on my laptop but actually playing the presentation from the computer. So they may be offset ever so slightly. But as usual, this is our review. So we'll start out where we started out after the last midterm, go as far as we get. And then, in the meantime, ask lots of questions, concerns, worries, etc. So we started out talking about lambda, and the really big deal with lambda, of course, is that we have this difference between lytic growth and lysogen formation, and then growth as a lysogen, i.e. integrated into the chromosome. And it's all about this regulatory region here in the promoter sequences. And PL and PR, mostly what we've looked at is what happens at PL, because that's what's regulating the first anti-terminator, the N anti-terminator, at which point when that gets turned on, you stop termination. That's the first round of stopping termination. The second round of stopping termination is turning on the Q anti-terminator, at which point you make all the late genes and everything lyses. On the other hand, we can have lysogeny. This is what happens if the host is unhappy. A couple people asked me about what do I mean by host being unhappy. That is that the cells are not growing particularly well, and the reason that the virus decides again to anthropomorphize to grow lysogenically is that there probably aren't going to be that many more hosts for it to infect. And so the strategy, again, obviously evolutionarily this has worked, is that if you think there are lots of hosts around, then you'll grow lytically, so you can infect more hosts, make more virions. On the other hand, if not, you want to grow lysogenically. And this is probably true for at least most of the viruses in the oceans because there aren't very many hosts around and there's not that much food around. So it makes sense that this is the process. The actual way that this happens for a lot of the viruses is completely different. We understand how it works in the terms of lambda, and it's all about the lambda repressor. Represses transcription from PL and PR, but stimulates its own expression, so positive feedback. And that's really, again, the way that you end up with lysogenic growth. Of course, the big question is how do you get C1 in the first place? Yeah?
So the role of N in determining lysogen as well is that if you have more N, it keeps the cell, it keeps the virus from going to lytic growth?
So the N protein, is going towards lysis. And you have to shut down the production of N in order to make sure you go lysogenic. And that's one of the roles of C1. So it's all about the production of C1 and really the beginning of the production of C1, which is what we actually, this is first just looking at that regulatory region. This is PR. PL is actually extremely similar. Regulation of PR, and again, PR is going to be expressing CRO and other things, but PL is going to be what's expressing the N protein. The C1 repressor, lambda repressor, will bind and block this, also again at PL. Eventually, there'll be enough of the C1 that it shuts down also its own expression. On the other hand, if you have the CRO protein will basically serve an exactly the opposite role. It will not repress PL and PR, and you won't end up with a whole bunch of C1. How do we regulate whether you have C1 or not having C1, this is basically the way that the virus senses whether the cell is happy or unhappy. If the cell is happy and growing really quickly, that means that there's lots of nutrients around, things are being metabolized, etc. The cells are very happy, and basically what that means is that you're never going to make, or at least sustainably make, C2, because C2 is what regulates C1, and that process is through C3. And C3 is really the sensor for the state of growth of the cell. So C3 will stabilize C2 if there is not many nutrients around. Okay. Then if you go down the C1 route, also what happens is you turn on the integrase gene, which will integrate the genome, and you will have this integrated form, again, which can persist over probably infinite number of generations as long as the cell is still around, unless you have induction, and induction is just the removal of C1, which then allows production of N and then the whole lytic cycle. Questions on lambda before we totally switch gears and talk about these RNA viruses. So, oh yeah, sure.
So just to
Andy.
clarify,
Yeah.
the more a cell grows, the more C3 is made, or the less C3 is made. The less C3 is made, the less that C2 is around, and that promotes. And the less C2 is around, the less C1 is around, and that doesn't end up blocking lysis. Is that basically how it works?
Yeah, so and this is just basically Andy's, right?
I'm Adam.
Adam, sorry. Close. Adam was saying that when the cell is growing well, you're not going to have very much C3, which will not stabilize C2, which won't turn on C1. And the way you can think about that is that, again, any of these mutants, if you have a mutant in C1, C2, or C3, you have clear plaques, that means you have lysis. So if you're missing any one of those genes, which is basically, again, what you see when the cells are growing happily, then you're not going to have any lysogen, everything will be lytic. Okay, more questions on these guys. And then the sort of virus from right field, left field, the fields, as the case may be. These plant viruses, again, this is a total oversimplification of all plant viruses. We're just talking about cucumber mosaic virus and the similar viruses that are related to it. The really big difference between these viruses and all of the other ones we've talked about in this class and are going to talk about in this class is that they use multiple capsids in terms of moving their genomes around. We've talked about segmented genomes. We just talked about flu on Friday. But those are all the segments packaged in a single virion. Here we have segments packaged in multiple virions. And that only works because the way that plant viruses get around is due to actual physical damage to the plants and huge inocula, huge numbers of infectious particles that get in, and statistically, evolutionarily, this seems to have worked. So that's one of the big deals, and particularly with these plant viruses. Yeah, Nicole.
So because they use multiple and they need all three,
Mm-hmm.
all three
Yep.
to make an infection, are those capsids considered virions, or are they just considered capsids? Not the whole thing.
Right. So Nicole brings up the wonderful point of whenever we try and name something, there's always going to be an exception, and it gets confusing. So would you call each of the capsids with genomes in them, say for cucumber mosaic virus, virions, or is the whole thing together then going to be a virion? Again, I would always just talk about this as being the extracellular form. So each of those would be a virion, and you happen to need 3 different virions in order to get a productive infection. But again, people might argue with me on that. So it's not the complete genome which is necessarily inside any one of these capsids to make it a virion. That was one of our discussions we had in the previous midterm was, what's the virion versus the capsid, et cetera. So the other important thing about these viruses that we really introduced here, but we see in many other of these RNA viruses, is what's going on at the 5' end and at the 3' end. At the 5' end, here we have caps, and the caps here are made by the viral protein, the methyltransferase. And as we heard about from flu, which again we may or may not get to given the amount of time today, those are all stolen. So the genome doesn't have to encode these capping enzymes. The 3' end, we have a difference with regular cellular messenger RNA in that here we have a secondary structure in the RNA, which is not your standard poly-A tail. Here it's a tRNA-like structure, and it's so much of a tRNA-like structure that the amino-acyl tRNA synthetases can actually put amino acids onto the 3' end. And this is great because 3' ends, one of the things that you need them to do is to protect against degradation. If you've got an amino acid at the end of your genome, then any of the hungry exonucleases are never going to be able to degrade it. So that's one way that these are protecting. It may be that this also helps translation. That's a little bit more of a controversial point about what these 3' ends in some of these plant genomes are actually doing. Does it stimulate translation or not? Would make sense because tRNAs, of course, are going to interact with the ribosome, and so that will help potentially to bring the ribosome to, to your mRNA. The other thing with these plant viruses is, yes, they get into a particular cell through cellular damage and then this massive inoculum, but then they have to get from cell to cell, and that process depends on these so-called movement proteins, which are really pretty specific as far as plant viruses are concerned. The main thing is basically getting from cell to cell. And this is pretty standard cell biology or plant cell biology, as the case may be. Plant cells are pretty tough. And so how you get from one cell to another has to do with these small, highly regulated, actually, junctions between cells, the plasmodesmata. Then the viral genomes are brought to these plasmodesmata by the movement proteins. And then eventually they can get into the circular system of the plants. And again, this is the process through these movement proteins, and it's a very active area of research exactly how these movement proteins work. But one inoculation, say you scrape a leaf of one plant, put your virus on it, pretend you're an aphid, that virus can then spread throughout the whole plant, and this obviously must be a very efficient process because you have to get all of these genomes segments to each of those other cells in order to get replication to take place as well. So the main reason to cover the, again, quote-unquote plant viruses in this class is to, A, give you a bit of an introduction. If you're interested, go to Oregon State, do a degree in plant pathology, learn all about these viruses, but also just in general introduce us to the RNA viruses, which we'll talk about for some of the rest of this. And then I can't not talk about this subject. This, of course, is not really a plant virus. It's a fungal virus. But the fungal virus that infects the fungus, which is endophytic inside the plant, which provides thermal tolerance. I think one of the coolest stories of the good viruses that clearly are necessary for the ecosystem. How many have heard about the term holobiont before? So the idea of a holobiont is it's all of the, one organism is actually not an organism per se, it's really an ecosystem of multiple organisms. You think about your microbiome, and then all of the viruses that are associated with that as well. So people are thinking about these definitions. You don't think about an organism, you think about the small community of organisms that are all associated in one particular place. And this is particularly true for animals. All animals have viruses associated with them, various different microbes associated with them, as well as the animal cells themselves. Then we finally get to talk about disease and first introduction to vaccines. Poliomyelitis was a huge problem throughout the world, and particularly in the US, and most of us have forgotten about it because of these two wonderful kinds of vaccines, particularly in the US, the inactivated polio vaccine or injected polio vaccine, either way you want to think about it, IPV, was developed by Jonas Salk in the mid-1950s. Basically, all it is make very large amounts of virus, inactivate it, and then inject it into people to get a nice immune response. This works very well, but it's expensive, and it doesn't spread, and it's relatively difficult to administer. You know, anytime you have to have needles, etc., means you have all kinds of other issues that go along with administering this vaccine. The oral polio vaccine is a version of the attenuated vaccine, and we talked a little bit about attenuation before. The whole idea here is that if you take a virus, infect something with that virus, take that virus again, basically do many cycles of infection, purification of virus in a controlled environment, you very often will end up with a virus which has lower virulence in humans. And sometimes it actually will be adapted to, say, the rabbits that you happen to put it into, or the mice, or the cell culture. There it's still pretty virulent, but in terms of humans it's not. Yeah? So we did not talk specifically about how it's inactivated. If I remember correctly, it's formalin. It's basically a chemical inactivation. But that I'm not absolutely certain about. But it's a chemical inactivation which leaves all of the antigenic parts intact. And it turns out that sometimes that works better than others in terms of these processes. Now, the live attenuated vaccine is great because you can actually use a lot smaller amounts of it because it's actually still an active virus. So it can replicate and make more of that attenuated virus. And then particularly in the case of polio, this is really useful because polio is not normally a neurotrophic virus. It normally replicates in the gut. And what it means is, particularly in areas where there's not great sanitation, that gets spread to people who haven't even taken the vaccine at all because they will inoculate themselves with the vaccine. The downside of this, of course, is that this attenuation process, all it is mutations, and those mutations can revert. So now, currently, on Earth, we have more vaccine-derived polio cases than we have of the wild polio cases. So we're going to have to switch back to using more of this IPV, which is a more expensive and more challenging vaccine to deliver, but it's not going to revert because you can't revert the, well, in theory, I guess you could revert the chemical changes, but highly unlikely to be able to. Oh, yeah, sure. Than wild cases, yes. Worldwide, yes. And the numbers are very small. They're on the order of hundreds, probably even less than 100 in some cases. Now, exactly how good the surveillance is in the places where these are happening, people could argue about that a little bit. Okay, more about polio disease. Oh, yeah, Jacob. For polio, I don't know exactly what the changes are. Again, they're mutations. I forget where they are. I think some of them may be in the iris sequence. I know that for some of the attenuated viruses, they're in iris sequences. But exactly where they are, I think there are 30 or 40 changes. I know for yellow fever, it's on the order of about 38 nucleotide changes. Interestingly enough, the yellow fever vaccine doesn't really seem to revert, whereas the oral polio does. Why? No one really knows. Okay, so how polio gets inside the cell, I should say how the polio genome gets inside the cell, is extremely similar to the way that bacterial viruses get their genome inside the cell. There's a pore which forms in the membrane after you have interaction with the receptors, and then the genome is injected through that. Probably also the way that you see with a lot of the bacteriophage. It's a high pressure because that genome is really packed inside the virion. As soon as you have an opening, that gets released. Yeah?
What's the receptor for polio?
The poliovirus receptor.
Oh.
Yeah, so it was found, in fact, before people were able to figure out what exactly it was doing regularly inside the cell. What it does, I can't actually remember. But yeah, the poliovirus receptor is the poliovirus receptor, also known as PVR. So yeah, again, you have this interaction. You have a conformational change that happens in the proteins, making a pore, and then you release the genome inside the cell. Now again, just like we talked about with those plant viruses, what's going on at the 3' end and the 5' end is interesting. And of course, the reason that this is the case is because all of these RNA viruses are going to be replicating in the cytoplasm. So if you're replicating in the cytoplasm, you have to have a messenger RNA which is recognized by the translational machinery, because so far no viruses have their own translation machinery. All of them are dependent on cellular translation. So this one's actually easier to start with thinking about the poly A tail. The poly A tail in polioviruses, and actually all of the, actually take that back, it's not all the picoRNA viruses, most of the picoRNA viruses, have these poly A tails, but they're encoded in the genome. So when that genome is released, again, after this conformational change, comes inside the cell, the 3' end has a nice poly A tail, poly A binding protein binds to it, etc. The big difference is, of course, at the 3' end, where you have VPG, which is a protein originally derived from the polyprotein right here, which is covalently attached to the 5' end through an OH on its tyrosine. So what else is in the genome? Again, mentioned this already, but it's a polyprotein. So it's one ginormous open reading frame that encodes multiple different proteins that get chopped out of this by, in the case of polio, viral proteases. And where do those viral proteases come from? They come from this polyprotein. What that means is that each of these viral proteases is actually active as part of the polioprotein. So they basically chop themselves out of the polyprotein, which to me is still kind of amazing. So those are the processes. You make this one big protein. It gets chopped into smaller pieces. You have all of the structural proteins here at the 5' end, the non-structural proteins at the 3' end. But since it's a polyprotein, they're all made at the same amounts. So there's no difference in terms of structural proteins versus non-structural proteins in terms of amounts of proteins that are made. Genome replication for these guys, I think I did a really horrible job of explaining the first time around, so I wanted to give myself another chance to do it. So if you look at a poliovirus genome, so again, we've got one up here at the top of the slide. Again, VPG bound at one end, poly A tail at the other end. Lots of secondary structures. This is also the iris, which is how the virus can have its genome be translated in the absence of having a cap structure. But these secondary structures which form in the RNA also are important for replication purposes. So one of the things that's important for replication is this 3CD protein. So 3CD, maybe I should back up here, is one of those polyproteins which is made through proteolytic cleavage right here, just after the VPG. 3C is the major cellular protease. 3D is the polymerase. 3CD, on the other hand, has yet another different activity. And that is binding here at the 5' end of the genome to some of these secondary structures. And as Dr. Hirsch mentioned in his talk about the flaviviruses, these secondary structures are absolutely critical for virus function. If you make mutations in the RNA here, you will find that the virus doesn't replicate anymore. So these secondary structures are very, very important. They interact with, again, this 3CD protein, but also cellular proteins. And this particular cellular protein, poly-C binding protein, give you one guess what it binds to. Lots of C residues. Also is critical for replication. These guys all bind at the 5' end. And then, just like you have with translation, there's a loop that forms between the 5' end and the 3' end. These guys are right next to each other. Just draw them out like this. And then you have the 3AB protein. Again, part of the polyprotein. This associates with membrane vesicles. And one of the things that happens when you have a poliovirus infection, you have this vast excess of vesicle formation. And it seems that pretty much all of the RNA viruses, particularly the picoRNA viruses, the flaviviruses, are all going to replicate at membrane vesicles. Why? Partly seems to be concentrating all of the various proteins that are necessary, but also in the cases particularly of the flaviviruses and then any of those other ones which are envelope viruses, it makes sure that your genome is next to where it's going to be finally made. So here, 3AB, 3A binds to the membrane. 3B is now the VPG protein. VPG protein, together with the 3D protein, has a couple of U's that get added to it. Why U's? Because they're complementary to the poly-A tail. So 2 U residues. These then serve as a template to make negative strand. This negative strand then also has 2 U's at the end, which will give you 2 A's. VPG, again, can bind to this and then make many copies here. One of the things that happens in poliovirus and all of these picoRNA viruses is that this negative strand formation is a lot less frequent than you have positive strand formation. So you end up with many more of the positive strands than you end up with of the negative strands. The primer for making these negative strands is that VPG and the tyrosine residue, which is on that VPG. So hopefully this is slightly more clear than the last time when I talked about it. Yes, no, happy, unhappy? So the primer is VPG, and it's the tyrosine on VPG. So we think about the structure of all these RNA-dependent RNA polymerases. They look a lot like DNA polymerases. DNA polymerases always need primers. What's the primer here? The primer is the OH, which is on VPG. So now Dr. Hirsch's lecture, flaviviruses. Flaviviruses are basically just like picoRNA viruses, only they have an envelope. Oh, yeah, in the back, sorry. Yeah, sure. Yeah. Yeah, so the question is basically, and for interest of time, I skipped over that here. So basically, what is shutting down vesicle transport? And basically, how does the virus make sure there are lots of vesicles around in order to make sure that they can replicate at all of those vesicles? I believe it has to do with the proteases, but I don't know what the targets are for that. Okay, other polio questions before we talk about the flavies? Okay, so again, these guys replicate at vesicles. In this case, it's the ER, and Dr. Hirsch showed this amazing TEM images of what these little things that seem to bleb off inside the ER and probably are also serving to protect the RNA while it's being replicated. The binding and entry of these flaviviruses, receptor-mediated endocytosis. There's a conformational change that happens due to the pH change. The genomes are released. There's a single RNA, which then gets translated, and you have a polyprotein. This polyprotein gets stuck into a membrane. And we'll take a look at that polyprotein in just a second. While it's probably already in these internal vesicles, you make negative strands of the genome, positive strands of the genome, and this then is processed through the Golgi. And the very final step, this maturation step, where you have a pre-M protein to the M protein, this is a proteolysis that happens now not from the viral proteases, it's a cellular protease. So that's one of the big differences that we have between these flaviviruses and the picoRNA virus. PicoRNA viruses is almost all going to be viral proteases. In the flaviviruses, there are a lot more of the cellular proteases, which we've also seen. We think about flu, where you get cleavage of the noodle, and where you have the coronaviruses, where we have cleavage that takes place. Most of these cleavages are going to be cellular cleavages, proteases, rather than the polyprotein cleavages, which are almost always going to be the viral proteases. Yeah, Nicole, you had a question?
Sorry, on the last slide,
Yep.
when it's the polyprotein,
Mm-hmm.
it's fusing with an endosome or a vesicle that's in. So where the
Okay, so so the polyprotein itself, that associates with the ER and eventually vesicles inside the ER. It's the fusion of the virion, the envelope of the virion, in the endosome, which releases the genome. And that genome then gets translated into the polyprotein. Yeah? The coronaviruses will also use some cellular proteases, so it's not just the flaviviruses. But the flaviviruses clearly use a lot of them, and we talk mostly about those. Okay, so this is also basically just the reminder, which I kind of talked about with the picoRNA viruses. Lots of secondary structures. Those secondary structures are very important for virus function. Also, what's going on at the 5' and 3' ends here. 5' end of all of these flaviviruses has a cap. Where does that cap come from? It comes from the activity of some of these viral proteins. Again, very similar to what happens in the plant viruses. The 3' end here also does not have a poly-A tail, has a lot of secondary structure, not tRNA-like secondary structure, but the secondary structures, they're proteins which bind to that and also protect the 3' end of the genome. And again, these secondary structures are absolutely critical for virus function. If you look at what's next to those, again, polyproteins with structural proteins at the 5' end, the non-structural proteins at the 3' end of the genome. What do those do? That's here. And again, this is that polyprotein as it's being translated. As it's being translated, it's being put into membranes and pretty standard signal sequences, which you would see with any typical secreted protein or one that's destined for one of these subcellular compartments. We have the E protein, which is the envelope protein. That's what's going to be interacting with the receptor. This M protein and the PRM protein, this is what's going to be that very last cleavage that takes place with the cellular protease as these virions are being matured. And then all of the NS proteins, the capsid protein is on the inside. That's what's going to be interacting with your genome once it's been made. And the big important protein here is NS5, which is not only an RNA-dependent RNA polymerase, but also a methyltransferase for making that cap, which we need in terms of getting cap-dependent translation to take place. Oh yeah, sorry, Adam. All of them, no. Important proteins to remember, I would say the ones that important to remember, like what I might be asking you on Wednesday, for instance. I would say capsid protein, E protein, M protein, and PRM. Those are the ones we've talked a lot about. I'd say NS5 we've talked a lot about. I would say NS4B. We have really not talked about that at all, so it's highly unlikely that it would be on an exam. ECMNS5. And maybe NS2B because that's a protease, a viral protease. Should probably just put together a list, right? Big table. Things to remember. So. Okay, so now I want to talk briefly about flaviviral diseases. The big ones are yellow fever and dengue. Important here is that these viruses replicate in the vector, almost always going to be a mosquito, and that replication is absolutely critical for actually getting disease. So you have to have a replication in insect before you can actually have a viral disease. So you can't get sick from somebody coughing on you if they have yellow fever. So that's the basic message here. Pardon? Yeah, so yellow fever is always transmitted by mosquitoes. The big difference in the flavivirus is hepatitis C, and that's still just blood, which is getting transmitted. But in this case, there are people who argue, and again, Dr. Hirsch knows way more about this than me, that a blood transfusion could get you yellow fever, but the cases there are very, very low. And whereas hepatitis C, pretty clearly you can. So it does really seem to be a mosquito-specific. Now, what that mosquito-specific step is, I don't know. And if I don't know, I'm not going to ask you about it on Wednesday. But that there are specific mosquito aspects of that. And then, dengue viruses, major issue with dengue is that it's spread throughout the world. But the major problem with dengue, particularly in terms of making vaccines, is this antibody-dependent enhancement, which Dr. Hirsch talked about, which is if you have exposure to one of the the 4 main strains of dengue and then are infected by a second one, often that second one seems to cause you much worse disease. And this is a big problem if you have an antibody response to, say, a vaccine, which doesn't cover that second virus which you've been exposed to. So that's the big issue with trying to develop vaccines for dengue. Lots of people are working on it. In fact, I just wrote a grant proposal together with my colleagues at OHSU to try and also work on some dengue virus and dengue virus vaccines. Hey, talk about the other question again you sort of had about the positive trans RNA viruses, these coronaviruses. We also wanted to talk a little bit about hosts and particularly reservoirs and reservoir hosts. And this is the case for yellow fever, for instance. The reservoir host for yellow fever is probably non-human primates and potentially other animals which are in what they call the sylvatic cycle of yellow fever. And that's partly why you end up keep having outbreaks of yellow fever. Not sure what the reservoir for dengue is supposed to be. I should go back and check. I think it's also animals and mammals. But certainly people were very concerned about the reservoir for SARS. And we now know that the reservoir for SARS is almost definitely the bat for a number of different reasons. One of them is because you have this very clear phylogenetic association of virus sequences with the human sequences, but also a vast diversity of these coronavirus sequences around that particular sequence. So it seems to have evolved from one of these, although these bootstrap values are complete garbage as far as I'm concerned. So back up. The other thing is that these bats seem to be perfectly happy with these viruses, and they can circulate, as far as we can tell, in bats basically permanently without causing any kind of disease. And so this whole idea of trying to find reservoirs for viruses is very important, of course, in terms of trying to control viral outbreaks. So what do you have in SARS? Also positive strand genome with a cap and a tail structure. Now, however, you have all of your structural proteins at the 3' end rather than the 5' end, and only, well, actually sort of 1 and a bit polyproteins. So this polyprotein and this polyprotein, sometimes you have a frame shift that happens here. When you're translating, this is always made as one messenger RNA because, of course, the genome is one messenger RNA. So it can be translated. Most of the time, translation will stop here. Sometimes translation will continue and go through the rest of this polyprotein. Just like all of the other polyproteins, this is mostly chopped up by viral proteases and has all of the various activities which you need, capping structures, polymerase structures, etc. I'm not asking you about all the names on all of these different proteins because I can't expect you to remember them if I can't remember them. On the other hand, here, and the big difference with these coronaviruses relative to the other positive strand RNA viruses we've talked about, is this set of structural proteins all present at the 3' end of the genome. And the way that, again, of these, again, I would have circled all of these things. I wouldn't remember what they are. I just remember the activities. So we've got transferases in terms of making cap structures, just like you have to have. RNA-dependent RNA polymerases, proteinases, proteinases. So what those names are, I would not remember because I can't remember what they are. So again, I just need to make one big table, right? Share it with everybody. How these structural proteins are made is really the, I think, fascinating part about these coronaviruses, and particularly, we also call them the nidoviruses, so nested subgenomic RNAs. The nido is for nest. What happens here is the RNA-dependent RNA polymerase binds to the 3' end of the genome, starts to replicate, hits these intergenic sequences, and then recombines, highly recombinogenic polymerase, either with the same RNA or with a different RNA, which is also present inside the cell. This recombination will jump to almost the 5' end, but not quite the 5' end, and replicate this short leader sequence. So jump, jump, jump. Very occasionally, it will make a whole copy of the genome to be your negative strand genome that you can eventually make positive strand genomes from. But most of the jumping is going to happen here at the beginning, which of course is at the 3' end of the genome. And you end up with all of these subgenomic negative strand RNAs. Subgenomic negative strand RNAs can also be bound to by the RNA-dependent RNA polymerase, which will make messenger RNAs for all of these structural proteins. And nested because each of these is nested within each of these smaller ones. So it's all about the recombination that happens while the polymerase is making a negative copy of your genomic RNA. And you're going to make many more of these just because the recombination will happen with the highest frequency here, next highest frequency here, next highest frequency here. Yeah? Yeah, so effectively, you're going to get a lot more of the structural proteins because you're making all of these messenger RNAs, and not even so much at the end. This will be very early on in the process. So basically, as soon as that genome comes inside the cell and it's been translated and you've made that RNA-dependent RNA polymerase, it's going to start making messenger RNAs for all those structural proteins. Yeah? Yeah, so basically what is this leader sequence? Yeah, so the leader sequence is just literally a sequence which is present on all of these subgenomic messenger RNAs and on the genome. And so this is how people figured out that this recombination had taken place. Oh, sorry, Nicole.
Oh, no. Are the leaders kind of like...
So are the leaders UTRs? Yes, the leaders are definitely UTRs. They also have all of the, now there's a cap here because these are all capped.
prior.
So the cap is prior to that UTR, yes. And also it's a binding site for the RNA-dependent RNA polymerase. Okay, vaccinate, vaccinate, vaccinate. How many times have I said that so far? Could say it some more. There, again, you cannot rule out that there is a very small possibility that there's adverse reactions to vaccines. There are some people who cannot get vaccinated, but particularly in the case of measles, it's a lot worse if you, like 1 in 1,000, 1 to 2,000 people who get measles will end up dying. Few in a million kids will get vaccinated, will have serious side effects. So Stedman loves numbers. Another way of looking at this graphically is as soon as the vaccine was introduced, the number of cases in the US went from hundreds of thousands down to thousands. And once we had a second dose, we had practically none. Of course, there are now reintroductions from other parts of the world, and that's where we have issues with this kind of disease. These measles and then all of the paramyxoviruses have this actually not unusual structure now as far as the negative strand RNA viruses are concerned. They've got fusion proteins generated by proteolysis, almost always by a cellular protease, making a fusion peptide, which then, in this case, it's a very, very small noodle attached to the F protein. In those other cases, often you've got much bigger noodles which are attached to them. And the paramyxoviruses are different in that they have a separate receptor binding protein from fusion protein. Most of the rabies viruses, so the rhabdoviruses, particularly like VSV, have just one protein, which turns out to be extremely useful for making cell fusion, getting your favorite recombinant virus to interact with whatever cell it happens to be nearby to, gene therapy, etc. One other thing that I think I did a crummy job of explaining the first time around, but it's also important because you see the same thing in filoviruses, but also in the orthomyxoviruses, is the binding of the N or NP protein, which is always on the inside of the RNA. So there's this helical structure that forms of the protein, and the RNA is wrapped around the outside. This, of course, only works if you have an enveloped virus because it's got to be protected somehow. So it's the envelope which is protecting it. In this case, the N or NP protein is binding to the RNA. It's protecting the backbone, but it's leaving the bases such that they can be made into messenger RNA or anti-genome, etc. And it is probably also protecting against exonucleolytic cleavage because the very last nucleotide is already bound to protein here. So, and then each of these 6 nucleotides is bound to one of these N or NP subunits. Again, depends on the various different virus that we're looking at here. This is nice because it always provides your template for the RNA-dependent RNA polymerase, which can now bind to this structure and then just replicate along here and never actually have to pull that RNA off of the protecting nucleocapsid protein structure. What do these genomes look like? Again, these are multiple proteins encoded in one genome, but this is now a negative strand virus, so 3' over here and 5' over there. The structural proteins are at the 3' end, and we'll see this also true for the filoviruses. These are made by the RNA-dependent RNA polymerase, which is encoded down here at the 5' end. Start, make a messenger RNA, get to one of the intergenic regions, stutter a bit, make a whole bunch of A residues, then often start again, make another messenger RNA, stutter for a while. In the case of PCV, you can also stutter in the middle of the open reading frame, which will give you a different protein because the messenger RNA now has an extra nucleotide in it. And just because of each of these intergenic regions, where you stutter with a certain percentage of the time, it will fall off. And that means you end up with more of your structural proteins and less of your non-structural proteins. How do you switch from genome to, or say messenger RNA production to genome production? As soon as you've made enough of your nucleocapsid protein, then you'll switch over, instead of doing all the stuttering and stopping and starting, to making whole copies of first your antigenome and then your genomic RNA. And this is true for the filoviruses. It's also true for the flu virus as well. Yeah? Yeah, so the antigenome is the positive strand from the negative strand, and then the genome will be the negative strand again made from that antigenome. Yeah, Adam, sorry. What's the mechanism whereby the presence of the N protein leads to the changes? It seems to be that the N protein is actually interacting with the polymerase, not so much as interacting with the RNA. Yeah, so it seems to change the activity of the polymerase, which of course can be a slight change in the structure. And the main thing that happens there is really you're not stuttering and you're not stopping. And so that's the main difference that seems to happen in the presence of the N protein together with the polymerase. Yeah. Yeah, if you're not stuttering, and stopping, because you also will stop and dissociate. Yeah. Yeah, at low N concentration. Yeah, well, the N protein is always bound to the RNA. So it's always bound to genomic RNA. Right, it will then make well, first your anti-genome copy, and then we'll make your genome copy. Yeah, back. Oh, so N is playing two roles there. So N is packaging your genome, so that's going to be clearly a structural protein, but also then serving as the signal to change the activity of the polymerase. And again, the same thing is true with filoviruses, and the same thing is true with the orthomyxoviruses of flu. So filoviruses, basically, again, just big rhabdoviruses, with the exception of the VP30 protein and the secreted glycoprotein. VP30 stimulating messenger RNA formation. Slight differences here because you've got overlapping starts and stops, but again, the main thing here is that secreted glycoprotein and yeah, and the messenger RNA processing, which is absolutely required for the structural proteins that you have in Ebola virus. I always promise not to make any kind of changes. I lie. This is the current outbreak, which is happening literally right now in the Democratic Republic of Congo. I think there have been 18 confirmed deaths so far, and unfortunately this is in a very remote location, so it's a little hard to get things there. So there are continuing outbreaks of Ebola virus in different parts of the world. Most people, of course, were concerned about the 2014-2015 because we had, I think the statistics I saw said about 7 to 10 times more cases in one outbreak than we had in all of the other outbreaks that people ever knew about between Guinea, Sierra Leone, and Liberia. Yes, we were really concerned about it. I still want to get the statistics on flu for this. And it was over 10,000 people who ended up dying from the West African outbreak. And finally brought under control by really strict, basically PPE, personal protective equipment processes, and investments by lots of NGOs, Médecins Sans Frontières, Doctors Without Borders, and the WHO. Sorry. Where did the outbreak originate? The outbreak originated in Guinea, right about here. So just right at the border between Liberia, Sierra Leone, and Guinea. That seems to be the first place. Now, how it got there from Central Africa, nobody knows. No, there's no real obvious way that it could have gotten there. There had been previous cases. There was an outbreak in Côte d'Ivoire, not that far away. So it may have sort of hop, skipped, and jumped. And then one of the big questions, again, having to do with reservoirs, is maybe it was a bat reservoir. And those bats had flown and had been transmitted, and then there was no cases. And then, for some reason, again, which is also not completely clear, there was an outbreak in that particular part of the world. A lot of people were just not expecting that disease there, and that was one of the main reasons that it spread and was such a problem. Yes? Oh, so how VP30 works, basically? Yeah. Right, that it overcomes the block to that, the formation of that particular messenger. So if you think about it, it's this start-stop process. So it's just like we had with measles and the paramyxo and rhabdoviruses. And so what happens in the case of, and when you don't have VP30 around, the polymerase just gets there and it stops. It doesn't make anything more. In the presence of VP30, then it can go through and make the messenger RNA. Yeah, so it just seems to hang out right there, and it blocks the RNA polymerase from coming through. Yeah, so so VP30 is in the virion. So when it first comes in, you're going to get the messenger RNA being made all the way through. And then if you have enough of the VP30, of course, it'll continue to make more and more. But if you don't have 'enough of the VP30, that specifically blocks, actually not even so much the VP30, but the N protein at the very, very beginning of the genome, so the 3' end of the genome. So that's where it's blocking. So it's basically sort of that first step where you're going to be making more messenger RNA. Okay, so the virus we should be much more concerned about, but only I think it was like 2 in 5 people actually get vaccinated for flu in any given year. Yeah, don't get me started on this. Again, nasty disease, which we won't talk much more about. This is that other segmented virus, which we're going to talk about in this class. There are other segmented viruses too, which we're not going to have time to talk about. But here, instead of each of those segments being in different capsids, they're all in one. How they all end up in one is still a very interesting question that I have yet to get a good answer for. We have the hemagglutinin, probably the best studied of the receptor binding proteins. Neuraminidase, which is the target of the best functioning drugs currently. are those that block the neuraminidase protein. Then we have matrix, as we've seen in many of these enveloped viruses. Difference here is going to be our ion channel, which is going to change the pH on the inside here. So first you have the change in the pH when you get into the endosomes, which causes this protein to undergo conformational changes. And then you have a change in the pH on the inside here. This is what used to be the target of a lot of those anti-flu drugs. On the inside, we have the NS2 protein. Again, what's a non-structural protein doing on the inside of a virion? And all of our genome segments, which have lots of nucleocapsid protein, which you can't see on here. But also all of the polymerase complex proteins, PA, PB1, and PB2. These all end up getting into the nucleus. Why the nucleus? Because that's where the caps are. And the way that this virus makes messenger RNAs is it steals caps from cellular messenger RNAs. So those caps are stolen through the activity of the polymerase. Those then are exported and then translated. All of these proteins are re-imported back into the nucleus. You make, when you have excess NP, that changes the activities of these proteins, just like the case you have with the paramyxoviruses. And those then start to make antigenome and eventually genome. When you have this genome assembled, now with the NS2 protein, this helps nuclear export, and eventually these guys getting packaged at the cellular membrane. NS1 is the only one which is not packaged inside the virion. This is the one which is blocking the cellular processes, particularly the processes making poly-A tails. If you don't make poly-A tails, you don't have fully formed messenger RNAs. Those don't get exported from the nucleus, so you end up with lots of unprocessed messenger RNAs with lots of caps on them, yum yum, and so the virus can steal more and more of those. And then protein kinase R, which is a cellular protective mechanism that actually Dr. Hirsch talked about really briefly when he talked about the flaviviruses, which is an innate immunity which basically says, hey, double-stranded RNA inside the cell, not good. We need to turn down all kinds of other cellular mechanisms. Finally, be concerned about flu. Don't be concerned about Ebola. And don't be too concerned about the midterm on Wednesday. Good luck.